Okay, we wanted to talk about parasitic weeds. These are the weeds which establish themselves on our crops. And it is quite a challenge because when we want to control these weeds, there's a possibility that we can end up also damaging the crop because they are attached to the crops. So the objectives of this lecture is to be able to identify parasitic weeds and their symptoms on the horse, to be able to understand the biology of the parasitic weeds, to be able to understand the general control strategies. So this is what we want you to learn. Here we've got an example of a parasitic weed, Striga asiatica, attached to grasses. Striga asiatica attached to maize in a field. This is actually in Gokwe. Asiatica on maize in the low veld. Striga asiatica on sorghum. Again, this is in the low veld. This is the red flowered plant at the base of this sorghum. Again, these are the effects of the Striga asiatica, although we may not see the weeds, but if you look carefully, you may find the plants emerging. This is the damage they can do. You can see the leaves are already drying, and it affects the photosynthetic activity of the plant, leading to low carbohydrate production and low growth. Here, it's actually, I think this is the maize planted in pots. We've got the parasites which are affecting the maize. You can see leaves are drying due to the impact of the parasite. Again, short plants due to Striga asiatica in Gokwe. Here it's a field where we have Striga asiatica. This is actually in Gokwe where we have farmers. This is in the field. Maybe you may see Prof. Gasura here with this is the student and farmers in the field. Again, Striga asiatica. We can have a variety of Striga asiatica, white and red flowered asiatica in this country. This is Striga forbesii, which attacks maize and sorghum on the heavy soils. Striga gesneroides, which can attack tobacco. This one is Striga emontica, which is found in Tanzania, Kenya, as well as West Africa, Nigeria, and the other countries surrounding Nigeria. We don't have this parasite in Southern Africa. Here is Striga emontica, again found in probably this is Sudan and East Africa. These Striga gesneroids in tobacco, again Striga gesneroids, Striga gesneroids in tobacco. This then is the tobacco which was affected by the infection of Striga gesneroids. This is Alectra vogleri with yellow flowers. It attacks bambara nuts. You can find this is the Alectra vogleri. Again, Alectra vogleri on bambara nuts. It also attacks cowpeas in Zimbabwe. You can see this is Alectra vogleri. This was actually evaluated in some of the experiments done by students here in pots. This one is Kescuta campestris. It looks like spaghetti with white flowers. It's a shoot parasite which is found on some crops. Again, Kescuta campestris. It looks like a spaghetti. This is the wiry sort of appearance. Again, Kescuta showing the flowers. It actually curls around the host. This one is Rhamphicappa fistulosa. It has got these trumpet-shaped flowers. It attacks rice grown in clay areas or wet areas of Zimbabwe. We have found this Rhamphicappa fistulosa parasitizing rice grown by farmers around Bushagashe in Masvingo area. Again, this is the Rhamphicappa fistulosa, which is already drying in a rice field. This is out of interest. You can have shoot parasites which attack trees. Gomarara, if you could have, you might have heard about the local name Gomarara. This is the mistletoe which affect trees. Again, the shoot parasite on trees. Here it's not yet a problem. But of course, we've seen it on trees, but it plays a role in the ecology of plants. If you want to read about it, you can read this paper by Hilton G.T. Ndagurwa with other authors. It's not a problem in the agricultural fields, but it's just out of interest that we do have parasitic plants which seem to play a certain role in the wild. This is the broomrape or bang species. It is found in Saudi Arabia, around the Mediterranean and areas, as well as Europe, North Africa. Probably it may be found in Ethiopia as well. So what are parasitic weeds? These are the weeds which obtain their carbon, mineral salts, and mineral salts from the host directly via the hostoria. We have examples which are the witchweeds. The local names are Bise, Ruombi, or Ison. Bise in Shona, Ruombi again in Shona. Isona is the better word for this parasite. So we can classify parasites according to their attachment on the plants, like this one. It's a shoot parasite because the parasite is found on the shoot. This is the host. If the parasite is found on the roots, it's a root parasite. Or we can use chlorophyll to differentiate parasitic plants. Hemiparasites contain chlorophyll and are able to photosynthesize during certain stages of their life cycle. For example, Striga asiatica. But holoparasites lack chlorophyll, which means they obtain their carbohydrates exclusively from the hosts. Now we've got an example which is Orobanchi. Now Orobanchi is found around the Mediterranean Sea countries and parts of Europe, as well as Saudi Arabia. It could be found in Ethiopia as well, but we don't have it in Southern Africa. Then Keskuta is somewhere in between holo and hemiparasitism, because it also contains a bit of chlorophyll. Now, according to Mbub in 1994, he indicated that about 73 million hectares, which were devoted to cereal production in Africa, were affected by parasitic weeds. So, in areas where cereals are grown in Africa, there's a potential of them being parasitized. Sobon in 1991 indicated that the grain yield loss in sorghum ranged from 6 to 21%. Kim had maize yield losses which were up to 40%. But it is also known that cereals and legumes affected by parasites may fail to produce grain. Here we do have parasitic weeds and their hosts. Number 1, Striga asiatica. It can parasitize maize, sorghum, millet and sugarcane. This also same applies to Striga foppsii. In Zimbabwe, we've got these two weeds which can affect our cereals. This one is mostly found on red soils, the Striga foppsii, but Striga asiatica is found on every soil type. Striga gesneroides affects tobacco and cowpeas, although we haven't seen it on cowpea yet. Then Alectra vogeli, it affects bambara nuts and cowpeas. And Cascuta campestris, it affects lucerne. Here I've left Ramphicapa fistulosa, which affects rice in wet areas. Now the plant family of the parasitic weeds, they are classified under the Orobangaceae family. Under this family, we have got Striga asiatica, Striga foppsii, and Striga gesneroides, Alectra vogeli, Ramphicapa fistulosa. Quite a number of parasitic weeds under that classification. Then we have one parasite under Cascutaceae, or sometimes known as the convolvulaceae. That is Cascuta campestris. Excuse me, let me admit somebody. I hope it's not going to disturb. Okay, someone is in now. Now here I've got a table showing the seed sizes of these weeds. There's no need to memorize them. We just want to show you that they are very small. Alectra vogeli has got okay, somebody wants to join this meeting. Let me try to admit. Okay, sorry for that. Right. And the weight of Alectra vogeli is 0.3 micrograms. Striga asiatica 0.33 millimeters length, 3.7 micrograms. Striga foppsii 0.5, about 12.4 micrograms. You can see that Striga vogeli has got the smallest size of weeds, of seeds. But all these seeds appear as dust, so they are very small. Now this is the seed production. Seed Striga Asiatica can produce 25,000 seeds per plant. Striga Forbesii about 58,000 seeds per plant. And these can last for 20 years being live. That is where the problem starts in terms of control. The production of seeds is very high and they can last for a long time within the agricultural fields. So this is the Striga Asiatica seed when looked under the microscope. Again, Cascuta campestris seed when looked under the microscope. If you see those dotted areas, this is where we find the Striga species. You can see in Africa, this is where we have a lot. Of course, they are in Asia, we do have some of them, even Australia, small bit in the US. In Zimbabwe, the Striga species or parasitic weeds are found everywhere across the agroecological zones or regions. Let us look at the life cycle of the parasitic weed. We should start with the seeds. Now, for the seeds to germinate, they need what is known as preconditioning. That is exposure to moisture at 25 to 35 degrees Celsius for 1 up to 3 weeks. Then they can respond to stimulants which are produced by the host. These germination stimulants are known as strigol. It could be sogolion, sogolactone, or alectron. But here I can say there are a variety of these substances which can stimulate the striga depending on the plants which are producing them. Now, once you have a germinated striga seed, it needs attachment to the root of the host. And for attachment to take place, it requires what are known as host initiation factors. One of the host initiation factors which was clearly identified is known as 2,6-DMBQ, which allows the attachment to take place, to form what is known as the hostoria. That is the connecting tissue between the parasite and its host. Now, at this stage, after attachment, the parasite can stay for a month underground, only to emerge later. And it is during this period when it does most of the damage. And the farmer cannot see it. So mechanical methods cannot be useful here. After emergence, you will have the plant having red flowers which are produced more or less after 4 weeks from emergence, and they then produce seed, and again the life cycle continues. So if I can make you recap on the life cycle, you start with the seeds, which need to be preconditioned. What we call preconditioning is the exposure to moisture and temperature ranging from 25 to 35 degrees for a period 1 to 2 weeks or 1 to 3 weeks. Then of course the seed is now ready to respond to chemical signals in the root exudates. This then is the germination of Striga asiatica. After preconditioning period, you get the radicles. This is under a microscope. Then you have attachment on the ostoria. So the germination stimulants which are produced by the hosts are called strigalactons. Please note that Strigor was isolated from cotton, although cotton doesn't support parasitism. We also have Sogolion and Sogolacton. Scientists have tried to mimic these strigalactons by producing Strigor analogs, GR24. Some scientists have tried to create herbicides out of these Striga analogs. You spray them in the field so that the Striga seed can germinate in the absence of the host so that it dies. We also have electrol, ethylene. These are natural products from the crops or plants. Then for contact with the host root, the parasite forms ostoria on the point of contact, but this requires what is known as 2,6-DMBQ or an full 2,6-dimethoxybenzoquinone. It's an example of the Ostoria initiation factor, HIF in short. Other products or substances, Ostoria initiation factors such as phenolics, flavonoids, cytokinins, thiodiazolone may also facilitate attachment depending on the host, but the one which is clearly identified is 2,6-DMBQ. Here you have the root of the host. This could be the parasite, and this is the bridge or the ostoria, which forms the connecting tissue between the parasite and the host root. Now the period underground, and now the parasite is very small and lacks chlorophyll, that is underground. It is fully dependent on the host, and the crop damage occurs at this stage, and it is not easy to control using mechanical methods because you cannot see it. It takes 30 days after germination to emerge. Now, 30 to 40 days after emergence, you get flowering, then you have seed formation, and the life cycle is completed. Now, let us wind up by looking at the effects on the parasite. Now the effects of the parasite are variable depending on the host. Sometimes you cannot see them. Maybe it's a tolerant variety or is a resistant variety. If the plant is susceptible or crop is susceptible, the host may actually die. So you have some cereals which are resistant or tolerant, whereas others are susceptible. That's why there is this variation in response. However, infected cereals, if the plant is susceptible, can have low stem weights and low grain yield. This is the effects of striga under field conditions. This is in Kwekwa. You can see maize was actually lodging. This is in Chundura communal area where we had planted some maize genotypes in striga hotspots. You can see here on this slide that the maize is wilting. These are the striga symptoms. Even if the field is not under moisture stress, there was plenty of moisture in this field, yet the plants were wilting. This is a clear indication of the presence of striga. This is SR 513, which is very susceptible under red soils. You can see the plants here are standard. Underneath there is a striga. This is the striga growing near plant stems. Now you can have standing of internode elongation. That's why we see short plants. Wilting symptoms, which I have indicated using the slides, which can happen even under wet conditions. Please note that there are similarities between the physiological effects caused by drought and those caused by striga. Now, striga tolerance is something to do with abscisic acid. Even drought tolerance is something to do with the abscisic acid. Now, striga can reduce the rate of photosynthesis in the host. You can have striga tolerant cultivars which are capable of maintaining photosynthetic rates. So, striga predisposes sensitive crop cultivars to photoinhibition, that is light impairment of photosystem 2. Striga infected cereals show lower stomatal conductance than uninfected plants. Now, if you have a cereal which is infected, it tends to close, to shut the stomata. Now, that closure will affect conductance. Where you don't have striga, the stomata will be open. So the presence of striga causes a closure of the stomata. And we can measure this using a porometer. We can also use portable modulated fluorometer, infragas analyzer. This can actually give us measurements on the photosynthetic rates as well as stomatal conductance. Striga can also cause phytotoxic effects on the host. Maybe there could be toxins, but these have yet to be identified. Now, if we look at the Alectra vogeri host interactions, Cascuta host interactions, there's a clear diversion of carbohydrates from the host to the parasite sink, and this can explain why the host is reduced in weight if you measure dry matter. Now, the parasites are very strong sinks for carbon and nitrogen, so they form extra sinks. But in a Striga serial interaction, the size of the Striga cannot account for the losses in carbohydrates. And in fact, parasite can make significant contribution to its own carbon requirements. But parasite biomass is just a fraction of the observed difference between infected and uninfected plants. So the question is, where are all the carbohydrates going to during infection? A carbon balance model indicated that only 20% of the carbon produced was directed to the parasite, so 80% was lost. Probably they were lost due to high respiration which was occurring, or it could be due to low leaf area and the low rates of photosynthesis maybe also account for these losses. Also, low stomatal conductance could be a contribution, could contribute to these effects. Like I said, respiration, or it could increase photorespiration metabolism that can contribute to a reduction in the rate of photosynthesis. There could be changes in plant growth regulators as a result of the presence of the parasite. So changes in host biomass allocation and architecture. For example, infected plants have higher root-shoot ratios and shorter internodes relative to uninfected controls. Changes in abscisic acid, gibberellins, cytokinins could cause architectural changes or the structure of the plant. And again, abscisic acid can cause low stomatal conductance. So this is it on the general introduction on parasitic weeds. What are they and what do they do? What is the life cycle of these parasitic weeds? So this is the general biology of the parasitic weeds which we tried to understand. Hello.
Yes, Doc.
Yeah, so this was it about the parasitic weeds. We want you to identify, you should be able to identify these weeds. You should be able to know their symptoms on the crops. You also need to understand their life cycle. Probably if we understand the life cycle, we can also get to understand the control methods which we can employ. So we are going to look at the control methods